Hello and welcome everyone to our webinar Legionella Detection, Culture Method or PCR. I'm your host, Orla O'Connor from BioProbe Diagnostics. And also with me today I have my colleague Dr. Elizabeth Minogue, Chief Scientific Officer and expert in Legionella detection and rapid diagnostics of microbial contaminants. We're delighted also to have with us today Sergio Marcus Estival, CEO and Head of the Microbiology Department at the Tentamus-owned Laboratorio Control in Spain. So before I hand you over to the main presenters, let me tell you just a little bit about who we are. So BioProbe Diagnostics is a startup. We're a spin-out from the National University in Ireland, Galway, and we develop solutions for the rapid detection of microbial pathogens associated with water contamination in a variety of different contexts in both environmental and industrial processes. In 2020, we received European funding alongside 3 industry partners in Spain and Germany to develop, test and bring to market a new qPCR test for the rapid detection of Legionella in water. BioLP-1 was born out of that project, and it has just undergone large-scale customer trials vis-a-vis traditional methods for detecting Legionella in water, and we're really excited to be able to share some of those results with you in today's webinar. So without further ado, I'll hand you over to Elizabeth.
Thank you, Orla. Good afternoon, everyone. I'm just going to give you a brief overview of our BioLP-1 product for Legionella detection and quantification. As many of you may know, Legionella species can cause a severe and often fatal form of pneumonia known as Legionnaire's disease. The associated mortality rates in the general population is approximately 10%. However, this rises to 25% when an illness is connected with a healthcare associated infection. The reported prevalence of Legionnaire's disease is increasing in many parts of the world. For example, in the US, incidence rates have quadrupled in recent years, while in Europe, cases have risen by almost 30%. The reason for this increase is largely due to an increased awareness of the disease, as well as wider reporting of infection, but there are other contributing factors such as an aging population, an increased number of immunocompromised patients. Other contributing factors include age and maintenance of buildings and plumbing systems, as well as global warming. It's also likely that these figures could surge even further as we emerge from the COVID-19 pandemic, due to an increase in the presence of stagnant water in buildings that were vacant during lockdowns, as well as an increase in vulnerable patient numbers. Unlike many bacterial infections, Legionella is not transmitted from person to person, but transmitted through contact with contaminated water. Legionella bacteria can be found in fresh water, however, can also thrive in man-made water systems. They can survive intracellularly in amoeba and other protozoan hosts that proliferate within biofilms. This can provide them with some protection from environmental stresses, such as disinfection procedures. Additionally, Legionella can rapidly multiply in stagnant water. For these reasons, it can be a problem in water distribution systems, such as those in hospitals, in commercial buildings and hotels. It's also a problem in things like decorative fountains and saunas, and obviously in air conditioning cooling towers. Because of this, there's a need for rapid, sensitive and specific Legionella testing to be able to identify potential problems at an early stage. This would enable immediate remedial action to be taken, which in turn could lead to a reduction in spread of infections and even mortality rates. To date, over 60 Legionella species have been identified. The leading cause of Legionnaire's disease is Legionella pneumophila. However, other species are also known to cause human infection. Studies have shown that Legionella serotype 1 could be the cause of up to 95% of all human infections. Therefore, any effective test should be able to detect and distinguish Legionella genus, Legionella pneumophila 1 to 16, and Legionella pneumophila serogroup 1. Traditional testing for Legionella using bacterial culture takes at least 10 days. This methodology is slow, and the testing procedure can be quite labor intensive. The turnaround time to result can lead to a delay in remedial action, as well as an increased health risk to the public from any contaminated water systems. Additional testing is also required to identify to the serogroup level. Alternatively, a real-time PCR assay for the detection of Legionella offers a much faster result. The test can be performed in less than 3 hours. This method is extremely sensitive and specific, and the multiplex capabilities of the technology allows for a full Legionella profile to be determined in one single test. Obviously, any real-time PCR assay then must be validated according to the appropriate ISO 12869. So this brings us to our developed technology, BioLP-1. The available test incorporates a DNA purification step, followed then by a real-time PCR assay. The DNA is purified from a filter and added to the lyophilized PCR reagents. BioLP-1 provides an internally controlled multiplex PCR assay. The 3 primary assays detect and distinguish between Legionella genus, Legionella pneumophila 1 to 16, and Legionella pneumophila serogroup 1. It offers a full Legionella profile in a single test. As I previously mentioned, the real-time PCR components are provided in lyophilized format, and they only require the addition of the DNA template to the sample well. So this reduces the risk of human error in the PCR setup, and it also means the product can be stored at room temperatures. The BioLP-1 PCR assay can be quantitative, allowing for the enumeration of the Legionella genome units present in a water sample. And this technology can produce much faster results, which can facilitate rapid remedial actions and potentially reducing the risk to human health. From a lab point of view, this test can help to increase lab productivity, allowing for the testing of a greater number of samples in a much shorter time frame. So these images are from a single real-time PCR experiment using our BioLP-1 assay. As you can see, there are 3 primary assays, as well as an internal amplification control, which is down here in the bottom right shown. The IAC here is used to reduce the possibility of false negative results, and it will indicate the presence of PCR inhibitors in a tested sample. So the 3 primary assays detect and distinguish between Legionella genus, Legionella pneumophila 1 to 16, and here Legionella pneumophila serogroup 1. In this particular experiment, a PCR calibration curve has been performed. Target DNA for each of the primary assays was added at a concentration between 1.5 by 10 to the 4 genome units and 15 genome units. So you can see the curves here. That one here is for 1.5 by 10 to the 4 on the left, and the lightest blue here on the right is 15 genome units. Each primary assay has a limit of quantification of 15 genome units per reaction, with the limit of detection being 5 genome units per reaction. The developed technology has been validated on both the LightCycler 480 and the CFX 96. However, it is designed to be open platform, and it should be compatible with most real-time PCR machines. Okay, so next up we're just going to show you a short video of a BioLP-1 real-time experiment setup. So if you just bear with me a second.
Good morning, everyone. My name is Sergio Marcos, and I'm CEO and Technical Manager of Laboratorio Control, laboratory belonging to the Tentamus Group. Next, I will talk about the realization of the comparative study carried out throughout this project. Within this work, there are two parts. On the one hand, the study between the results of qPCR rapid method and the ISO 11731 culture reference method of Legionella analysis. On the other hand, a study of life cycle analysis and cost-benefit analysis has been carried out between the two techniques, which I will talk about at the end of this presentation. To make the comparison, water samples have been collected in duplicate and different facilities, varying the matrices. All the samples collected have been analyzed by two techniques, culture by ISO and by qPCR bio Lp-1. Around 6,000 samples have been analyzed for each of the techniques, and the results obtained have been evaluated at 75%. We wanted to reflect in this study the reality of matrices that are recorded in the laboratory routinely, and therefore a high percentage are water for drinking water, followed from the cooling tower and the swimming pools. Raw data of three types have been obtained: A. Culture and PCR positive, B. Culture negative and PCR positive, and C. Culture positive and PCR negative. Each of them has a very relevant importance even when both positive techniques have come out. The logarithmic difference between the counts has been made. These are the results obtained for each of the evaluation based on the percentage obtained in positive samples in Legionella species, Legionella pneumophila and Legionella pneumophila serogroup 1, both in those that are the two positive techniques and in the one that is only positive PCR. The total count of Legionella predominates the Legionella pneumophila and finally Legionella pneumophila serogroup 1. In the case of the positive culture and the negative PCR predominates the Legionella serogroup 1 followed by the Legionella species. It should be noted that the cases found at this point are with a very low number of colonies on the plate, with very low statistical reliability according to the ISO 8199. Other studies and requirements for analysis: different biocide and different neutralizing have been tested, and for this, samples have been continued to be collected, but a spike kit has also been made on other microbiology bottles left over from the laboratory in order to have more matrix. Different species, Dumorphy, Anisa, Migdae, or something like this, different biocide, different neutralizers. In addition to these analytical studies, Laboratorio Control has evaluated and reported in order to issue its conclusion. A comparative study between the two standards involved life cycle analysis and cost-benefit analysis, a study of different international regulation. The opinion of our clients has been collected on the results they have been obtaining, very important for the study, and continuous feedback in order to improve the protocol. Response from customer trials. The traditional cultural analytical techniques studies requires many test plates and highly experienced technicians capable of both carrying out the Legionella test and also the reading and final interpretation of the results. And the culture method has the fundamental problems: long incubation time, which delays the action between 10 days and 14 days, impossibility of detection, variable non-culturable, and low recovery rates, and very important, low specificity, interference by accompanying microbiota. I want to show you the following photographs, which have a large number of other non-Legionella bacteria, which after a rapid growth, 2 or 3 days after incubation, and despite having inhibitory treatments and um, of- for their growth and selective culture media mass detection, not being able to give results in the laboratory. In these cases, qPCR is especially useful because it does not interfere with the detection of Legionella. While real-time PCR has the capability to provide fast and robust results and is highly specific for Legionella, while also being very sensitive, being able to detect the presence of Legionella growing in a water system before it becomes a serious health risk, and avoiding the high economic costs for the companies that an outbreak can cause. Both the economic and the environmental costs are much higher when Legionella water testing is done by culture than when the analysis is done by the real-time PCR method. Very important in a private laboratory like us, who has ISO 14001 quality and environmental management, as well as a great efficient and environmental commitment. Thank you so much for your attention. I leave my email for any question or consideration you want to consult me. Thank you so much.
Thank you very much, Sergio. So BioLP-1 now available on the market through our official supplier in Germany, Genexon Bioscience. So we've come to the end of the webinar. We've got a few minutes for questions. Eliza, we've had one question come in here. Can you tell us about the specificity of the assay? What Legionella species are detected?
Okay, thanks, Orla. So the specificity of the multiplex PCR assay was tested against a large panel of microorganisms. These were both Legionella and non-Legionella species in accordance with ISO 12869 for Legionella detection. At least 40 species of Legionella and strains must be tested for both inclusivity and exclusivity. So we've tested over 60 Legionella species and strains, with the results being 100% specificity. Additionally, we've tested the assay against at least 20 other non-Legionella microbial species that are commonly found in water samples. And again, the results were 100% specific.
Okay, thanks, Elizabeth. Next question. What type of DNA isolation and purification method would you recommend?
So DNA purification components, they're all supplied as part of the BioLP-1 Legionella kit. The water sample would then be filtered and the DNA can be lysed directly from the filter. The DNA isolation and purification kit is a column-based DNA extraction, and our BioLP-1 real-time PCR assay has been validated using this DNA extraction methodology.
Okay, we've time probably for one more question. Can the presence of multiple Legionella species in a water sample be a problem? So can this affect the assay performance?
The presence of multiple species of Legionella in a sample should not pose any problem for the assay. In the past, we have performed a series of experiments to challenge the assay in just this manner. So in these experiments, we were able to detect low concentration of Legionella DNA in one primary assay, for example, 5 genome units, while in the presence of a very high background of another one of the primary assay targets. This background was in the region of 1 × 10⁷ genome units, so very, very high background. These experiments showed that the limit of detection of the assay, the limit of quantification, and in fact the standard curve were not affected by having multiple targets present and by having them present at very high concentrations.
Lovely, thank you, Elizabeth. Unfortunately, we're running out of time. We do have some more questions, but we will reply to each and every one of you individually. Thank you so much for your questions. Thank you all for attending today. And just to remind you, today we have recorded today's session and we will send this out by email to everyone who's joined us today. Thanks again, and bye-bye.